That's what I want to do. Okay, so we should be recording now, and then this will be uploaded. One thing before we get into today's topic, I just wanted to share my screen and show you what I've posted in EduCat. So under the readings and videos, hopefully you can see my screen now, EduCat. There's several of the top readings are just how to go through the process of reading a scientific paper, and these do kind of repeat themselves. I'm not asking you necessarily to read all of it. Pick one that works for you, but I think it's important because a lot of what we're going to be doing in this class is we're going to be learning through the published scientific literature, and so learning how to read that kind of writing is going to be to your advantage. For myself, what I typically do when I read a scientific paper is of course I read the title, then I read the abstract. Typically the next would be the introduction, and then I skip to the end. I go to the discussion and conclusion section. And then once I've done that, I go back into methods and results. And so typically that helps me have a better framework of what they were trying to accomplish. And so when I get to the more dense methods and results section, it's a little bit easier for me to process. And so I find that I almost always do that method of, the intro and then the discussion first before I get into methods and results. I do that often when I'm reading a paper that's out of my discipline, where I'm reaching a little bit and I'm not as familiar with the topic. So I would encourage you to check these postings out to get a feel for that process. And then the actual readings that I showed you yesterday are right here with the evolution topics. Okay, so please start reading that. I will be posting 2 other articles on evolution and microbes. And I will say up front that one of them is more written by geologists. So obviously I'm not going to be asking you to learn all the geology of what they're talking about in the paper. There are several key points that they make, big picture points that they make that I think it's worth looking at. And then there's a second paper I will post that's actually a transcript of a talk that was given by a Nobel laureate on microbes and evolution. And so it was his talk, and they published it. And so that one again is some of the details I'm not going to be asking you to understand to any significant level, but some of the big picture ideas I think are well said. So I will post those shortly. But so before we also, I'm going to make sure that I'm doing this right. So now I'm going to switch to my PowerPoint. And if at any time I'm sharing my screen and I'm saying, now you should be seeing this, and you don't see it, please let me know. So I don't go on and on and on, and there was nothing there for you.
A quick question. Are
Yes.
these PowerPoints going to be available for us online?
Yeah, they should be in EduCat. I think these are the ones I'm going over right now are already posted in EduCat. Look under the PowerPoint study guide section in EduCat, and they should be there.
Okay, thank you.
Yeah, absolutely. So I just wanted to touch base on the study habits for this class, and pretty much any class that you are working through at the college level in particular. And for lack of a better descriptor, I call it repetitive studying, and that sounds really boring, and I don't mean it to be, but it's fairly accurate idea where you're trying to look at the material several times before you walk in on exam day and actually take the exam. And so this is what I learned my sophomore year as an undergraduate when I was taking organic chemistry and physics and genetics and I think it was an English writing class. And I was stumbling in organic chemistry. I utterly failed the first two exams. I think the first exam I got a 36 out of 100, walked out of there in tears going, what the heck is going on here? And so when I was meeting in office hours with my genetics professor at the time, Dr. Verley, he asked how my classes are going. I said, I'm utterly failing organic and I don't know what to do. And he's the one who taught me how to study, which is, and this was a while ago, but the neuroscience is currently showing that this still works. And so this idea of getting at the material more than once, but you do it in short bursts of time. You don't sit for hours on end and try to study. You study for 20 minutes to a half hour, and then literally you stop, get up, walk around, take a break, shift your brain into thinking about something else for a little while, and then you come back into studying after a 15-minute break or something. And if you consistently study, once I learned how to do this, it was basically I study for short bursts of time every day. And then at the end of the week, I would review my notes. And so, we meet 3 times a week for lecture. So even on the days I wasn't in that class, I would still study that class. And so every single day I would study for short bursts of time and then review at the end of the weekend. When Dr. Verley told me to do that, and he said, give it a shot, let me know what happens after you take your next exam. My next exam happened to be in physics. I took the exam and I actually kind of panicked because it was almost too easy. And I was one of the first people done, and that never happens. I used to be always the last person done, where the professor was often like yanking the test out of my hands. So I kind of panicked and went, I must have done something wrong. I walked out of there, got the exam back a few days later, and I couldn't believe it. I got a 98 out of 100. That never happened to me in physics, ever. And so I was sold on this idea. It worked. And it really also helped ease my stress level. When I walked in on exam day, I felt much more prepared. Obviously, attending lectures and labs is really important. I feel that what you gain in lecture when we're in person with each other synchronously and going with the lecture makes a big difference. And putting things in your own words. So I do provide PowerPoints, but also reframing things and rephrasing things in your own words is important. That's part of the neurological process of embedding things into your long-term memory. And obviously assigned readings are going to be important, especially in this class. And so, I talked to a lot of students over the years that say, I really studied hard for this exam. I don't know why I did so poorly. And believe me, I get that. I've been there. And I learned that there's a difference between studying and effective studying. And effective studying, at least for me, and I think for a lot of our brains, many people, it's this idea that you can't just kick back, put your feet up, crack open the textbook and start reading and think you're studying. It's a process. When you study effectively, it's a process where you have to be very involved and engaged in what you're trying to learn. And the way I started doing that is trying to make things very personal to me, where I could link it to something in my life that either I thought was funny or silly, or I had an emotional feeling toward, where I could link it to something important in my life. And that often was the trick for me to get things to sink into my long-term memory. Even like silly little things, like I'm sure you remember from elementary school or middle school, you had to learn about the rainbow and the colors of the rainbow, red, orange, yellow, green, blue, indigo, violet. And if you spell that out, and just use the first letter of each of those colors, it's R-O-Y is red, orange, yellow. Well, that spells Roy. That's my dad's name. And so that was very easy for me to remember. It's very personal, right? It's my father. And then the next initials are G-B-I-V, or green, blue, indigo, violet. Well, in my family and my dad, he loves the Green Bay Packers. So GBIV is Green Bay is victorious. And so it was, again, very personal to me. And to this day, it was so easy for me to learn that. So when I got asked, what are the colors of the rainbow, and then, which colors, which wavelengths of light are the longest wavelengths, I got it, I knew it. And so again, making it personal and emotional really helped me learn something like that. And so I try to do that all the time. I try to make things silly where I'm cracking up, making myself laugh, maybe no one else gets the joke, but if you can infuse learning with laughter and silliness, it actually helps you to learn the material. The neuroscience is proving it to be true. So effective studying, I will be talking a little bit more about that over the next couple of weeks. At the start of every lecture, I'll give you some tips and ideas to think about. But
Quick question.
yeah, go ahead.
So for the repetitive studying, when you said you would study for like 20 minutes and then you would take a break, how long was like that break period time?
Usually around 15 minutes.
Okay.
And it was enough for me to get up. I used to study when I was an undergrad. I'd go to the library.
Mm-hmm.
And I used to, walk around and run up and down the stairs and just get my oxygen pumping again, so I wasn't sleepy and more, get the blood flow up to the brain. And so I usually do something physical, go for a walk or run the stairs or something. And then if I studied, if I studied at home, sometimes I switched to studying in my room, because sometimes I needed personal things around me, because I would actually link what I'm learning to certain physical things in my room. Like I collect rocks, polished rocks. I just love the beauty of polished rocks. And so I'd have rocks, and I'd have, other things that, gifts that were given to me that I had an emotional attachment to, and I could link what I was learning to things in my room. And that was also a trick that was really effective for me when I was learning things. And I like to move around too. I would often move around when I'm studying, and for some people that works really well, just that physical movement. So you'll probably see, if we ever get back to face next week, I can't move around so much this semester because I need to do Zoom recordings, but I typically, even when I'm teaching, I move around a lot. And so if that works for you, if that's a good strategy, try it out. I'm encouraging you to try things out and just see what you think, because your undergraduate career, you study a lot, and you might as well make it efficient, and you might as well make it fun, because so much of your life is devoted to it. Make it fun, make it silly. And it does decrease your stress when you feel like you're on top of it and you're learning it. It really is amazing what it can do to decrease your stress level. Okay, more on that next week probably. But I wanted to just try to get you thinking about the history of microbiology in the kind of the grand scheme of things, and to kind of get it in framework of, when did this all begin, and what was the overall progression of this field of study. And, because most microbes are microscopic, people didn't know about them for the longest time. And in fact, when people way back when in the 1600s and previously were proposing that there's things that we can't see with the naked eye that are affecting us, of course, a lot of people scoffed at that and said, you're crazy. And so I would love to go back in time and be a fly on the wall and see these time periods and just see the progression of how, eventually people started to realize that this is actually happening. There are these microscopic organisms. And so back in the 1600s was when it finally gained traction with people that, yes, there are these organisms. Fungi were easy to see with the naked eye. Protozoa, not too bad if you had a halfway decent microscope. But it wasn't until a little bit later in the 1600s that bacteria you had to get a much better microscope, and Leeuwenhoek was the dude that was credited with that, where he could actually see bacteria under the microscope. And then it wasn't for another couple hundred years that finally this idea of microbial ecology, or ecology in general, I should say, actually started to be thrown around and thought about as a field of study. And so then if you combine those terms with microbial and ecology, it's, just looking at the relationships that microbes have, both with their physical surroundings and with their living counterparts that they're interacting with. And so that really didn't gain serious attention until about the 1960s, believe it or not, which isn't really all that long ago. And so, and I think a lot of that has to do with just the lack of technology. It was really hard to ask good microbial ecology questions that you could actually put to the test, because the technology just wasn't there yet. And so the 1960s is when it really started to gain traction. So here, just looking at this very, very simple microscope that you see in the middle here, and Leeuwenhoek developed, and then some of his drawings, you know, very simple drawings. And I love the picture with all the hair there, the wig. And so, you know, this is, again, it's just amazing to think about the idea that, you know, microbes were here on the earth first, and yet we didn't know of their existence until the 1600s. A long time of them really dominating the earth, and we had no clue. So anyways, this is another person that you'll hear about in lab a little bit more, but Bijorink is someone who got into the study of plants and looking at how microbes and plants interact with each other. And he's the one that figured out how to do what we call enrichment culture, which you're going to learn how to do in lab. And it's basically the idea of trying to target certain microbes. Like, you know, if you take a soil sample and try to grow microbes, you're going to get all kinds of microbes out of that soil sample. But if you say, I want to grow microbes that are really good at eating a particular carbon source, well, then you add in that carbon source and you've enriched it, right? You've enriched that media, and you're going to get a certain subgroup of microbes that can eat that particular carbon source, for example. And so, and believe it or not, you're going to be working with agar plates, and you're going to learn how to make agar media and pour that into agar Petri plates. And even that, you know, having agar, that was a huge breakthrough in the field of microbiology, because they couldn't find a substrate that you could grow microbes on top of without the substrate turning into a liquid goo, because the microbes always broke it down. Microbes are so good at breaking down all kinds of different nutrients that no matter what they tried, and you know, you want a hard physical substrate to be able to look at these microbes and study them. And it took forever for them to figure out if, so when you look at agar plates, the actual agar that hardens up and solidifies everything, that's actually extracted from red algae in the ocean. And they still do that to this day. And most microbes cannot break down that agar substance that the red algae make. And so that was a huge breakthrough. And anyways, so enrichment culture is just playing with the recipe of, you know, what kind of nutrients go into that media in order to target certain microbes. He also studied the sulfur cycle. So when you talk about microbial ecology, you do get into biogeochemistry, which is just looking at how the microbes break down matter and recycle it. And so, you know, you talk about the sulfur cycle, the nitrogen cycle, the carbon cycle, and so on and so on. And so Bijorink was way into the sulfur cycle. And he actually was, did some pretty decent work with viruses of plants. There's a virus called the tobacco mosaic virus. And so this was back in the 1800s into the early 1900s that they were making some progress in understanding microbes and their impact on Earth. And then Winogradsky, we'll also learn about him in lab, because the Winogradsky column that we'll set up as one of our experiments is obviously named after him. And he was also really good at studying soil microbiology and learning about the types of microbes in the soil through enrichment culture. And again, looking at the nitrogen and sulfur cycles and how the microbes are absolutely critical for those biogeochemical cycling processes. This term is, I'm going to come introduce this again more than once, chemolithotrophy. Of course, I know it's a science term, so they got to make it big and long and hard to pronounce. But chemolithotrophy is looking at this idea of, we're going to learn how microbes are can be incredibly diverse in how in what nutrients they can use in order to gain energy and get their ATP. Chemolithotrophy means that they're actually using inorganic nutrients as an energy source and an electron source. And we can't do that. Humans just absolutely cannot do that. So litho actually means rock. When you look that part of the word up, it means rock. And trophy means to use or to eat. And so they're if you kind of look at that word, it means rock eating. They eat rocks, which is trying to say that they can take inorganic nutrients, minerals, and use that for an energy source and an electron source. And microbes, some microbes are extremely good at that, and there's a lot of diversity in how they do that, and we'll get into that a little bit. And those of you who've had Gen Micro with me, that word should sound familiar, I hope, after a summer of not thinking about microbes perhaps. Autotrophy, this is another thing that microbes are amazing at. Plants can do this too, but microbes were the first to do it. And so autotrophy is the idea of they can make their own sugars. And plants, as we know, can do that through photosynthesis. Microbes can do their own version of photosynthesis. And then there are also microbes that can do autotrophy through some other means, not like how plants do it. And so we'll touch on that a little bit too. So anyways, Sergei Winogradsky, he was really good at, early on starting to pick apart these ideas and starting to put together a framework of knowledge in these different areas. And so we'll set up a Winogradsky column and we'll learn more about some of these processes going on. It's really cool. It's like a self-sustaining ecosystem all in this big, huge test tube that we'll work with. And over the weeks, we'll figure out what's going on in there. So another topic that's kind of within the microbial ecology field is remediation, and it's a topic that's not going away. In fact, it's just getting stronger and stronger because of all of our pollution efforts that we're doing over the years here. So in the 1970s, that's when it really came into the forefront, looking at, realizing how much we've polluted our environment and that we need to try to address this. And because they knew that microbes were so good at breaking down all kinds of different compounds for their nutrition, they started using microbes back in the 70s for remediation. And we'll talk about this toward the end of the semester. There's some really cool work going on in bioremediation. So when you hear the term bioremediation, it's usually what they're thinking about is microbes, using microbes to break down pollutants. There's another term called phytoremediation, phyto, P-H-Y-T-O, phyto. Anytime you see P-H-Y-T-O in a word, that means plants. And so phytoremediation is the use of plants. And so there's several terms that have been coined over the years. But this is still a hot topic and still a very useful idea. And then, of course, in agriculture, we need to eat. We need to feed billions of people. And it's becoming more and more of a concern of how are we going to do that we are running out of usable agricultural land. It's pretty much all used up already. The other land that's available is either too droughty, too salty, too polluted. And so how do we feed all these people? Well, now they're starting to turn to the microbes. There's a big push in the agricultural scientific community that we need to stop with all the pesticides and herbicides, and we need to look at how plants and microbes interact and figure out ways to capitalize on that to really boost plant growth, plant defenses, and to improve the soil. If you don't have good soil, you don't have good plants. And so microbes in the soil are absolutely crucial for the soil fertility. So we'll get into that as well through the semester. And this is taken from some textbook, microbial ecology textbook. And again, I'm not asking you to memorize any of this. It's just having you try to get a feel for some of the kind of the big picture events that happened in the field of microbial ecology over time, and just getting familiar with some of the ideas that have been played around with.
I have a quick question.
Yes.
Are we gonna get examined on who did what for microbial ecology, or we're just going over this just to know?
Yeah, I'm not going to ask who did what. Dates, general dates, like knowing it's the 1600s is kind of where things really started to happen. That I would like you to appreciate, to put it into perspective, but I don't really get into who did what ideas. You can easily look that up. You don't have to memorize it. So, but dates I think are important to kind of get your head set on that.
Okay, thank you.
Yeah, absolutely. Let's see. Another thing we're going to learn to appreciate in this class is anytime you're looking at ecology, you tend to have very complex systems that you're studying. And, you know, when I took like BI 210, the Principles of Ecology course, they didn't even really get into microbes, at least back when I took it. They probably still don't. They probably don't have time to even get into that. There's so much other stuff to talk about. And so when you look at ecosystems, and, you know, and you look at the macro ecosystem of the plants and the animals and, you know, everything like that, and then you start layering in the microbial impact as well, it gets hugely complex. My nephew right now is studying at the University of Vermont and working on his PhD, and he's looking at forest ecosystems, and he's also thinking about the microbes in those systems and how they're influencing carbon storage in the soil. And his data is just incredibly complex that he's getting, and he's at that stage where he's writing his thesis and analyzing his data and getting a little frustrated. And I keep trying to tell him, you know, it's because it's so complex. There's no easy answer to what is driving these processes going on in the ecosystem. It's incredibly complex. And so I think you're going to quickly start to get a feel for that as we work through this semester, that there's a lot to think about when you look at microbial ecology, especially in the framework of ecosystems, complex ecosystems. And again, another thing I hope to drive home pretty quickly and throughout the semester is that microbes are so incredibly fundamentally important in ecosystems. And unfortunately, you know, a lot of other classes you may take, they just don't really talk about the microbes. And so I'm trying to, through this course, have you better appreciate that microbes are everywhere. They are heavily involved in so many of the processes that you've already been learning about in your classes at Northern. So we'll get into that. So again, just looking at some of the ideas here with the biogeochemical processes. So microbes have been doing biogeochemistry for, depending on who you talk to, if you have a chat with an evolutionary microbiologist, they would throw out dates around 3.5 billion years ago. Some push it to 4 billion years ago. Microbes were here on Earth. They were cellular at that time, and they were actively influencing what's happening on Earth. And in fact, one of the papers I'm going to post for you to read, they are throwing out the hypothesis and suggesting that microbes not only were impacting like the gas structure of our atmosphere, they actually were involved in the formation of the crust and the continents of Earth. It was through their action that the physical solid substrate that we call Earth, you know, the crust, and it's really an interesting hypothesis. It's a hypothesis. You know, it's kind of hard. They're working on it. I don't know if it'll ever be fully put into a theory mode, but it's an interesting idea to think that the microbes were that fundamentally involved in literally shaping Earth. And so we're going to get into that a little bit more. Again, just showing complexity, looking at just one idea of nitrogen, you know, how is nitrogen cycled through? And I guess, and again, I'm not going to ask you to memorize this right now. This is not, you know, this is just an intro. We'll come back to this later in the semester. I'm just trying to kind of throw out these big picture ideas right now. And if you look at some of these words that are written on their end here, denitrification, nitrification, ammonification, those are all microbial actions. Those are microbes doing their thing. Here on the upper left, you see microbial nitrogen fixation. That's an incredibly important process in the cycling of nitrogen. And microbes are the only ones that can do that. So nitrogen fixation, if you've not heard of that term, that's the idea of taking nitrogen in the atmosphere. So we know our atmosphere is about 80% N2 dinitrogen. microbes are the only ones that can take that nitrogen gas and fix it into a usable form that can go into a physiological reaction. Plants can't do it. The animals can't do it. It's only key bacteria that can do that. Fungi can't do it either. Fungi technically are microbes, but they can't do it. Only bacteria can do it, and it's only some bacteria that can do it. And so we'll get into some of the biogeochemistry as we work through the semester. But again, I put this here in this set of PowerPoints just to get you to appreciate that just in this one cycle, when you look through it, microbes are incredibly important in the whole process of the nitrogen cycling. So we'll get into that a little bit more. So most of the time when we say ecology, when you hear that word, most of us think about macroecologists, where people go out and they study certain ecosystems, forested ecosystems perhaps. And so technically they would be a macroecologist. They're studying the big things. So if you're a macroecologist, you go out to the field and you go to these different sites and you are looking at what's going on, mostly with the naked eye, and you don't really do much lab work. And that's fine. I mean, that's just a general description of a macroecologist's life in their career. Microecologists, on the other hand, they also go out to the field and they have to both be a macro and a microecologist, really. They go out to the field, they have to observe what's going on, they take samples because they're looking for the microbes, and then they bring it back to the lab and often do intensive lab work. And so when you look at the idea of the microbial ecologist, it's really fun because you get to do both. You get to go out into the field sites and interact with organisms in that manner, but then you also get to go and play in the lab with the samples that you take and try to tease apart what's happening. We'll talk a lot about this in the class and what are the pros and cons of that, what are some of the pitfalls you have to look out for, because anytime you take microbes out of their element and bring them into the lab, there's a lot of things you need to think about in terms of if you're going to set up an experiment, to really have that experiment give you answers that are accurate to what you're trying to learn. And so we'll get into that a little bit more. So here, looking at this idea of, okay, let's go out to the field site. So on the top of this table here, you're looking at, whoops, that's not what I wanted. Go away. Sorry. All right, the microbial processes going on in the field site. So you're trying to figure out, okay, at this field site, what's happening in the soil or maybe in the water system. And so you need to take samples and bring them back into the lab. And so commonly what you would do then is, especially if you think back in the 1960s and 70s, what they would do then is try to grow these microbes up and get them purified out from each other to try to study them better. Like, who are you? What are you doing? What's your physiology? You know, if you can understand a little bit more of how are they working in that ecosystem in terms of their physiology. And then, of course, as technology advanced, we could get more into, well, what kinds of gene expression patterns are they showing? What genes are turned on and off? What are those genes coding for? And so you could get into the genetics of that. And then eventually the technology got better for molecular biology questions, where you could really get into the nitty-gritty of what's happening at the cellular level. And that's all great. I mean, it's great to learn about your microbes and all of that, but then you have to be able to think about, well, okay, what are they doing back in that field site, you know, from an ecology perspective? And you have to be careful, because what can be happening in the lab doesn't necessarily mean that's what they're doing out at their field site where you got them from. And so that has not really been a question we could answer very fully until really recently, when we had a much better technology, the ability to sequence genes and DNA. And so the technology has just within the last decade or so gotten to the point where we can really start teasing apart, okay, what are they really doing at their field site? You know, it's one thing to see what they can do in the lab, but what are they doing at the field site? So we'll get into this more too. Again, this is just an introduction to get you thinking about you know, how microbial ecologists have to do their job and try to tease apart what's happening in these different ecosystems. So there are lots of different disciplines within microbiology that microbial ecologists will interact with. And now more than ever, all sciences really are getting much more collaborative. We're much more willing to reach across the table, so to speak, and to be much more interdisciplinary, because we're learning the value in that, and that we shouldn't just be off on our own. And so I'm glad to see that. There's a lot of different ways to approach a question you're trying to answer, and having more people involved in it, the better, usually, in terms of the different expertise that they can bring to the actual experiment. So again, just trying to get you to appreciate that especially at the microbial level, there's a lot of different ways you can approach answering these questions in terms of the scientific experiments that you're going to do. This last term, omics, is fairly new, and that is looking at, you might have heard proteomics, genomics, transcriptomics, metabolomics, and so we'll talk about those terms as tools essentially for how to figure out what microbes are doing, and so we'll get more into that as well. Just listing more again, just to get you thinking about all the different disciplines that can be involved in microbial ecology. Okay, so microbes have been on the face of the earth, depending on who you talk to, 3.5 to 4 billion years ago. So they are thought to be the first living organisms on Earth. And when you look at the fossil record, although it's really hard to find good fossils of bacteria, that's for sure, they are there. They have been able to find them. And they do resemble bacteria. When you look at the fossils and study them carefully, we'll talk a little bit more about that. But when you're thinking about the evolutionary process, really it's just, change through the generations. It's usually is how it's often talked about. And of course, Charles Darwin with the idea of natural selection, that, the habitat that you are engaged in and interacting with will influence how those organisms evolve and speciate. And in general, when you think about the idea of natural selection, one of the topics that's often heavily discussed is this idea that, DNA mutates, it changes. And spontaneous mutation can occur, chance mutations. And then if that mutation happens to be beneficial for that organism, where that new trait that they evolved works well for them in their particular habitat, they will maintain it and they will pass that on to their offspring. And if you can imagine that happening over time, you could see where speciation would slowly, you'd start to see the separation of organisms into different species depending on the natural selection processes, the habitats they're in, and how those mutations play out in that organism. But I want to really emphasize that when you're looking at evolution, you have to put it into context of the habitat that organism is in. So for microbes, if you take a microbe from the soil environment and pop it into the ocean, it probably won't survive. Even though that soil microbe might be ideally suited for the soil, they don't have the right repertoire of genes to be able to be hugely successful in the ocean. And so it's all about their current habitat. And if that mutation, that chance mutation occurs and it's favorable, well, then they'll maintain it and pass that on to the next generation. As that happens, you can get, speciation to occur. And so this idea is something that I want you to think about through this class, because whenever we discover a microbe doing a novel thing that we, it's like, oh, that's new. I didn't know they could do that. Again, framing it in the idea of, well, why are they doing that? And going back to the idea of evolution and natural selection and thinking about these processes really helps you to better understand why microbes are doing certain things. And it also helps you to think about applied uses of microbes too, if you can keep that framework of the processes of evolution and how that influences the microbe. Okay, I am going to switch. Questions on anything right now as I'm switching? Whoops, it's not what I wanted. So I'm going to share another set of slides. And I'm assuming you can see it unless someone tells me otherwise. So this PowerPoint slide set is posted as well in EduCat. And the second quote there is something, again, the point I'm trying to drive home is, if you can keep that lens of evolution and the processes of evolution and speciation in your head as you're learning this material, it will make more sense. And so Dobzhansky is a Nobel laureate, I believe, and this is attributed to him. In biology, nothing makes sense except in the light of evolution. And so that's why I like to talk about these ideas early on, especially in my micro classes. And really driving home the point that microbes were here first. They've been here for billions of years. They have changed the earth and continue to change it and alter it and structure it in a way that allows for other organisms to live here. And so this timeline, I would like you to think about the 3.5 to 4 billion years ago. That time stretch is, I think, an important thing to keep in your head in terms of remembering numbers, because that's where they're finding the evidence showing up now for the formation of cellular life and microbes showing up, right? So of course, we can't go back in time and actually witness this, but there's a lot of scientists that are working on this idea. And certainly they all seem to agree that, way back when, our early Earth was very different than what it is now in terms of the geology. And again, that paper I'm going to post, now they're saying they believe microbes could have had a profound impact on the geology of the Earth, as well as the atmosphere. But early Earth's atmosphere didn't have oxygen, for example, that it was rich in CO2, no oxygen. Oxygen was poisonous back then. And so, we had carbon dioxide, water, ammonia, methane, hydrogen. Those molecules were important and prevalent, but oxygen didn't show up until the right types of microbes started to go through photosynthesis and start pumping out oxygen. And so you might have heard about Stanley Miller's experiments in the lab, where they created an experiment that simulated, some of what might have been going on in early Earth, with looking at, heat and an energy source, gases. They would mix the right kinds of gases and then, apply heat, and they would look to see what would show up in the, when they drew off the water and looked at what kinds of chemicals might be in that water after this experiment was finished. And they were actually able to find very simple amino acids had formed under these conditions. So starting with just, inorganic chemicals, you ended up with organic molecules. And so that when Stanley Miller, and I believe Stanley Miller was a master's student at the time when he did this experiment, and he was not, he didn't already have his grad degree. So it was a big deal when this came out. And so from his experiment, they got a simple amino acid. Glycine is one of the simplest amino acids. And, they would repeat the experiment and they would get these other amino acids, and they're like, holy cow, we're actually generating organic matter from simple inorganic matter and applying an energy source. And so that kind of took off. Then a bunch of people started getting into it and going, wow, what else can we do? And so then they started realizing under the right conditions, you can make not only amino acids, but other organic matter, nucleotides, ATP, which of course, that's how our bodies run on that chemical energy source, carbohydrates, phospholipids. So all of this is organic matter that's necessary for the organisms to exist. And so then they started going, well, how could this be happening? And where could this be happening? Where on Earth do they think, early Earth might have really sparked and started to happen and form cellular life? And when I was an undergrad, the current idea was, they think it was along the shores of the ocean. That's what they were getting into back then. And then they realized maybe not. And so they started looking for surfaces, clay surfaces that had the right minerals, especially iron. And the surface of clay has the right kinds of mineral structure as well as the charge structure to allow for polymerization to occur. And so they started proposing ideas like that. But they were still saying maybe along the ocean. Well, then that lost favor. They still like this idea of polymerization. They actually talk about, soup. So you have like the ocean water with all these minerals and chemicals, and under the right conditions on a clay surface, they could polymerize and form more complex structures, which would be the crepe idea, like a pancake. And so they got into that idea, and they still think that that's a process going on, but now they've shifted where this might all have happened way back when. And so they're pointing now to the deep ocean and in the hydrothermal vents of the deep ocean. And so we're talking about the ocean floor. And that paper I'm going to post, that's where they're zooming in again. And actually, they're not only just talking about the ocean floor, they're talking about what's happening beneath the ocean floor. And so this is something that they're getting into. Now, I'm not going to get into, you have to learn the whole processes here, but looking at this idea that there's minerals here at the hydrothermal vents, there's incredibly hot water coming through there, incredibly hot, so energy source, and you have clay and physical rock structures that could allow for formation of more complex molecules under the right conditions. And so now they're pointing to the hydrothermal rocks and vents in the deep ocean, that's where early Earth might have actually arisen. What time do we have? My clock doesn't show. Ooh, my clock doesn't show with when I have my, okay. Let me just say, we're gonna get into, I'll summarize a little bit more on Monday, I guess, a little bit more about the formation of cellular life, and then we'll start getting into the next topic. But does anyone have any questions right now?
I'm just curious, next week is already in person, right?
I'm hoping Monday we're going to be in person. So look for an email from me over the weekend. I'm waiting to hear from Northern. I don't know. I don't know yet. So they haven't announced anything yet. I'm sure they're going to do that soon. And then we will plan for that. And then I'll say who's going to be in person and who will be Zooming in. Okay.
Okay.
Well, just check your emails. I'm sure there's going to be a flurry of them from Northern and from your different teachers. So, okay, I think that's it. So I'll see you tomorrow though for lab. We'll get into lab Zoom at 10. I don't think it's going to take the full 2 hours. I don't anticipate that, but I need to talk to you about lab and how we're going to function in the lab. And then I wanted to talk to you about the field guide and the research project a little bit. So I'll see you tomorrow at 10 for that. Okay.
Okay.
Have
Bye.
a good rest of the day, everybody.
You too. Bye.
Thanks.
Thank you.